Pleasure to introduce you. Tony's known Larry for a long time, and I think that applies to quite a large number of the speakers at these events. Nevertheless, one thing you should know about Tony, and if you're an immunologist, you need to know these things too, and that is that there are these numbers called CD numbers, and cells have a large number of them from CD4, CD8, CD69, CD83. This guy actually knows what they all mean, and that is something, I haven't met anybody who actually knows that, coupled with which that he actually knows some genetics as well. So coupling understanding those cell surface antigens with genetics is truly it's a pleasure to introduce him, and he's going to tell you a little bit about autoantibodies to DNA and its importance in lupus. Tony.
Thank you, Tim. So it's a bit early to be clapping, right? So you need to hear it first. So I want to thank Larry. I have known Larry for a long time, and I want to thank him for seeing through this wonderful idea for this symposium to the current fruition. It's awesome. I also want to thank Meredith and Janet and other people in the foundation who've made sure that this process works smoothly and make sure that all of us do what we're supposed to do when we're supposed to do it. So I want to talk to you about lupus from the perspective of an immunologist who's been interested in immune development and how that contributes to the potential for autoimmune disease, how genetics may contribute to autoimmune disease, and then finally, for the disease lupus, why it's so heterogeneous and why that heterogeneity makes the disease so difficult to diagnose and to treat. So first of all, lupus can be benign and can involve only the skin, which is illustrated here with these lesions that can be painful and annoying, but they're not life-threatening. But lupus can also be systemic, and systemic lupus erythematosus, as the name implies, is an autoimmune disease that affects many tissues and organs in the body, including the skin. This is the characteristic malar butterfly rash that's photosensitive over the bridge of the nose and the cheeks. Arthritides, so that individuals with lupus most commonly have arthritis, but the most potentially life-threatening aspect of lupus is the potential for the autoimmunity to cause glomerulonephritis. And so the loss of kidney function can lead to death in lupus because the kidneys fail to function. Drug-induced lupus is also a phenomenon that's well known, and the drugs that are listed here are well known to be able to cause lupus, and they do so because they're able to modify histones. They each share the characteristic of being able to alkylate histones, and that process makes the histones immunogenic. And so as long as the drug is present for individuals that are susceptible, then there is systemic lupus erythematosus in those individuals. You take the drug away and the disease goes away. All right, so here is the demographics of the disease. In the US, it's present in about 20 to 150, depending upon the part of the country. In the UK and Europe, it's about 95 per 100,000. China, about 70 per 100,000, although that estimate is probably way low. It's more predominant in women than men, and that predominance is in part contributed by hormonal effects due to estrogen. It's more prevalent, and the disease is worse in people of color, African Americans, Hispanics, Asians, and Native Americans, than in Caucasians. It's also more common in urban than rural areas. And interesting, this interesting little tidbit here, it's not just a trivial point, but it points to the fact that there is probably an environmental component to the development of lupus, and that in individuals with lupus, their pets are more likely, dogs are more likely to have lupus than other people, And so maybe it's a component in the environment that is contributing to the development of lupus. So the clinical symptoms of lupus are listed here. Malaise and fibromyalgia, chronic inflammation. Its neurologic and psychiatric manifestations of lupus occur in many individuals. The highest mortality in lupus is due to cardiovascular disease, but the highest mortality ratio is due to glomerulonephritis. And so, clumsy. So the expected deaths from glomerulonephritis are much higher than would be expected than in the general population for individuals with lupus, pointing to the effect of this autoimmune disease on the kidneys. So lupus was first realized around 916 AD, with Heterogenesis described a disease of the skin in which there were lesions, erythematous purpurous lesions, that across the face, that to him looked like the bite of a wolf. And that's how it got the name of lupus. That name extended into the Middle Ages, up into the mid-1800s, when a group of dermatologists in France, led by Cazenave, wrote a manual of skin diseases in which they first described the disease lupus erythematosus. It's an erythematous rash of the skin in individuals with that disease, lupus erythematosus. Up until that time, it's thought that maybe lupus erythematosus was caused by tuberculosis, just a sequelae of the infection. But it was determined that that's probably not true by Kaposi and von Hebra, who said that no, this skin disease is unique. It's not due to tuberculosis. It's a unique skin disease, and it was von Hebra that first described the butterfly rash that I showed you earlier, in which you get this photosensitive rash on the cheeks and bridge of the nose in the shape of a butterfly, hence the name. In the early 1900s, up until the 40s, there was a Canadian physician named William Osler who described patients with lupus erythematosus that also had nephritis. And so this was the first indication that maybe systemic lupus erythematosus was more than just a skin disease, and that it could involve other tissues or organs. And the most important of these is the glomerulonephritis in the kidneys. And so the inflammation indicated here, and changes to the basement membrane of the capillaries in the kidney that prohibit proper filtration, which is what leads to the loss of kidney function. It wasn't until the late 40s that systemic lupus erythematosus was determined to be an immune disease, an autoimmune disease. And that occurred because Hargraves discovered something in bone marrow prep from a lupus patient called the LE cell. This is a polymorphonuclear leukocyte that had engulfed the nuclei from another cell. That was further substantiated by the finding of Henry Kunkel and his colleagues in the 50s that the LE cell was dependent upon a serum factor, a protein, that bound to nucleoprotein, and that it was this factor in the serum that promoted the formation of the LE cell, and that actually the LE cell didn't exist unless the sample, blood or bone marrow, was allowed to sit before it was observed microscopically. And so it was due to this factor binding to nuclei and then promoting its engulfment by phagocytic cells that form the Ellie cell. In the early 60s, Brown and colleagues described lesions in biopsied kidneys from lupus patients, in which they described these subendothelial and subepithelial electron-dense deposits that Kunkel and colleagues later determined were IgG autoantibodies to DNA and chromatin. And so the characteristic of glomerulonephritis that occurs in lupus is due to the presence of IgG autoantibody that deposits within the glomeruli. And this is just an immunofluorescence of a kidney from a patient showing those IgG deposits. All right, so why autoimmunity? Evolutionarily, you think that the immune system has had a long time to get it right? To devise a system that should be able to eliminate pathogens, but at the same time would leave us alone, leave your own tissues alone. It's a long time to solve that problem, but it didn't solve it. We haven't solved that problem. So why? Well, if the premise is that the immune system is there to protect us against pathogens, then you think about, well, what does it have to do? Pathogens are extremely diverse. They replicate really, really quickly. They diversify and evolve very quickly, far faster than any host that they might want to infect. And that's true of us. So the evolution of the immune system, both phylogenetically and ontogenetically, has been to maximize the potential that you detect and eliminate a pathogen at the expense of the potential that there may be autoimmunity. And with the evolution of that potential to detect pathogens, there has been coevolution of regulatory mechanisms that are intended to control the immune response so as to prevent a response from a pathogen from getting out of hand and causing inflammation, or from the ability of immune cells to respond to self. So, if we think about how the immune system works, normally we are infected and the pathogen is detected and then eliminated and we're fine. That requires two types of immune response. An innate immune response that's dependent upon cells like polymorphonuclear leukocytes and macrophages to detect the presence of a pathogen and to start to affect its removal, its elimination. In the absence of those cells, not only do we not eliminate the pathogen, but we don't initiate an adaptive immune response. Innate immunity is required to initiate an adaptive immune response. In the presence of innate immunity, functional macrophages and polymorphonuclear leukocytes, we start to eliminate pathogens, but in the absence of the guidance that is necessary from adaptive immune cells, we still fail to eliminate those pathogens. So we have to have both. We have to have the innate immune response to initiate the process and to activate adaptive immune cells, which then come around to focus the innate immune system to be better at eliminating the pathogen. Okay. So, chance. It is this incredible potential of the immune system to respond to pathogens that creates the chance that autoimmunity may occur. And so let's take a look at how that chance is manifest. For innate immune receptors, they're inherited and and phylogenetically conserved. They are so-called pattern recognition receptors. They recognize things that are unique to pathogens that shouldn't be on our cells. They have very limited diversity because they just simply recognize pathogens as a group. It's just, they're pathogens. They're a virus or a bacterium or a fungus, but not a specific type of virus or bacterium or fungus. That's the job of the adaptive immune system, that specificity. Most somatic cells express the same pattern recognition receptors. They all detect the same things and evoke the same type of response. Those determinants that are recognized, the pathogen-associated molecular patterns, are, for the most part, conserved and unique to pathogens. But those pattern recognition receptors also recognize something called DAMPs, damage-associated molecular patterns, or danger-associated molecular patterns. And it's that aspect of innate immunity that creates the chance for autoimmunity. So PAMPs, examples of those are bacterial products, cell wall products like lipopolysaccharide, endotoxin, RNAs and DNAs from viruses, bacterial DNA. Bacteria don't methylate their cytosines, and so the innate immune system through toll-like receptor 9 uses that as a tool to be able to recognize that a bacterium is present, either outside or inside of a cell. Damage-associated molecular patterns are indicated by this group of molecules. These are interesting because they should all be inside of a cell. They should never be outside of a cell. If they're outside of a cell, that means there's a problem. And the innate immune system detects those molecules as an indication of danger, of there's something wrong, and so we need to mount a response to this, just in case. These are just byproducts of damage to connective tissue. And uric acid, that's just an unfortunate formation that causes a lot of pain to some people. But uric acid, it wasn't until recently that we understood why uric acid would induce inflammation. And that's because there are a group of pattern recognition receptors called nod-like receptors here that can detect uric acid and initiate an inflammatory response. So, PAMPs and DAMPs are able to activate pattern recognition receptors, toll-like receptors, Rig-like receptors, nod-like receptors, and DNA sensors. And they all initiate signaling through the innate immune system that will either affect pathogen elimination if they're PAMPs, or if the response is to DAMPs, the outcome may be chronic inflammation. And so hence chance. The innate immune system has inherent chance for the response to self. It just has to be regulated. Adaptive immune receptors are not inherited as such. We inherit the potential to make these receptors. We inherit genes that can be randomly recombined somatically to generate individual receptors. Every lymphocyte has a unique receptor, which means that the diversity of that set of receptors is enormous. But that carries with it an interesting problem. First realized by Carl Landsteiner, who discovered the blood group antigens. Along with discovering blood group antigens, Carl Landsteiner discovered that he could immunize animals, rabbits, with small organic compounds attached to a protein. And he could induce an immune response to those compounds. And that immune response was exquisitely specific, so he could distinguish dinitrophenyl from trinitrophenyl. But what that meant And what he realized was that the immune system has not evolved specifically to detect pathogens. It has evolved the ability to detect anything. And it's that diversity which creates the chance for autoimmunity. We discovered in mice and Heidi Waterman in humans that up to 50% of the potential to produce antibodies in mice and humans can be autoreactive. 50% of the pre-immune potential can react with nucleic acids, with DNA. So that means that that potential has to be regulated. It has to either be eliminated or controlled. And in the absence of that, then everyone has the potential to generate an autoimmune response. And I think this experiment, done by Dharmesh Desai in my lab many years ago, illustrates this point pretty well. We took normal mice that normally do not make antibody to DNA, and we immunized them with complexes of an immunogenic peptide bound to DNA, along with an innate immune stimulus in the form of an adjuvant. And in every mouse we immunized, they developed antibody that could bind to DNA. This represents the serological response of autoimmune mice, spontaneously autoimmune mice, that naturally produce antibody to DNA. So those mice that were immunized were really quite similar to autoimmune mice. The antibody that they produced not only bound to DNA, but they could bind glomerally and could induce glomerulonephritis that we could detect as kidney damage due to proteinuria. Here are mice. This is the kidneys from mice not immunized. The kidneys from mice immunized peptide alone. So what this said is that, well, DNA can be immunogenic under appropriate circumstances. The chance is there. There's always the chance that one can make an antibody to DNA. And that once that antibody is made, that's all you need in order to initiate disease that resembles systemic lupus erythematosus. The fact that the response is regulated is indicated here, because with each boosting immunization, we stimulated the production of anti-DNA antibodies in these mice, but if we waited several weeks, that response declined and went back to sort of pre-immune levels. So what that meant is these mice were immune, but they weren't autoimmune. They had the potential, but they weren't autoimmune because the potential to produce antibodies in those mice was regulated. Genetics. Genetics in lupus is really complicated. We know that there is a pattern of inheritance because the sibling ratio is pretty high in lupus. And so here is the population frequency for siblings just in the normal population. This is the sibling ratio. This is the sibling risk for individuals who are in a family in which another sibling has lupus. And so this high ratio here indicates, well, okay, there is a component of inheritance to lupus. But what is that? And that's where it becomes complicated. Because by and large, if you examine by GWAS, genome-wide association studies, what genes make one susceptible to lupus, that for most genes identified, there's a really modest effect size. So most of these genes are in some way or another involved in immune responsiveness. But look at that. It's a very modest effect on the potential to develop lupus, with exceptions, right here. And that exception is interesting, because what do these things have in common? They all have in common that they're important for the elimination of cellular debris. The elimination of nuclear and cellular debris. TREX1 is an endonuclease that digests DNA in the cytoplasm. The complement components that you see there are important because they bind to and direct cellular debris to phagocytic cells for removal. If those are inoperable or don't operate at the optimal levels, then there's a potential for lupus. And so for TREX1, if you get that mutation, it's rare, very rare, but if you get it, you're going to get lupus. So now to heterogeneity. And here is the problem with this disease, and why it's such a terrible disease, because of its effects, but also because of the complications that it creates for diagnosis and therapy. So here are the criteria that are used to diagnose systemic lupus erythematosus. These are immunological criteria, different autoantibodies. These are clinical criteria. And when you look at this, there's nothing there that's unique to lupus. Those are chronic inflammatory conditions that are common to many different rheumatological or systemic autoimmune diseases. Any 4 of these, with at least 1 from each category, constitutes a diagnosis of lupus. That is remarkably heterogeneous. And it makes it very difficult for a clinician to have a diagnosis that they can be really absolutely confident in. If an individual has nephritis and autoantibody to DNA, that's enough. Then they have lupus, and that can be treated. All right, so we move on to look at actually the heterogeneity of the autoimmunity itself. Individuals can make a lot of different autoantibodies in lupus, the majority of which are directed to 1 or more nuclear components. And they're illustrated here. The component that I've always been the most interested in, the autoantibody, and the autoantibody for which there is the highest clinical correlation to the manifestation of glomerulonephritis, which is the most severe, I would argue the most severe manifestation of lupus, is to autoantibody to DNA. But yet, within that, there are a group of patients that are referred to as serologically positive, clinically quiescent, who have very high titers, serum titers, of autoantibody to DNA, but who never develop glomerulonephritis. So why is that? Is it because autoantibody to DNA is not necessary to induce glomerulonephritis? Or is it that there's something about the way that the antibody induces the disease that can manifest as that difference? So how do these autoantibodies cause the disease? As circulating immune complexes that lodge in the kidneys. This is what Henry Kunkel thought here. Is it because chromatin can deposit on the glomerular basement membrane and antibodies can bind to that and initiate nephritis? Or is it possible that the autoantibodies just bind in the kidneys by themselves? So to answer that question, which has been the question that has occupied my interest for a long time, we took advantage of autoimmune mice. These are New Zealand black by New Zealand white hybrid mice that spontaneously develop an autoimmune disease that looks like lupus. They make high titer autoantibody to DNA, starting at around 3 to 6 months of age. By 10 months of age, they're all dead because they have glomerulonephritis. As part of our interest in trying to understand this, we made these autoimmune mice transgenic for a single monoclonal autoantibody to DNA called 3H9. That autoantibody binds to DNA perfectly well. All of these transgenic mice produced very high titers, serum titers of autoantibody to DNA, but not one developed nephritis. Go figure. And so here it is. Here we try to create this. We went to a lot of expense, actually, to create these mice that we could use to study B-cell development and disease more easily because all of them are making exactly the same antibody to DNA. They didn't get sick. The wild-type littermates all got sick. They all developed nephritis normally. So why is that? So again, the question, do these antibodies really cause nephritis, or is it something else? If they cause nephritis, how is it? How is it they do it, and why didn't 3H9 cause the nephritis? So if we think about how the antibodies bind to DNA, DNA is acidic, and anti-DNA antibodies, as a rule, have basic amino acids that are part of the antibody combining site. And those basic amino acids and other amino acids can form ionic or hydrogen bonds with phosphate oxygens on the DNA backbone, as is illustrated here. And all this is a model that's probably pretty correct because we're looking at B-form DNA there, if you take, and which is A's and T's on each strand alternating. If you take a strand of A, strand of T as double-stranded DNA, that double helix is compressed by about 1/2 base pair per helical turn. Those antibodies do not bind that molecule. And so we know something about the specificity of the antibodies. We also know that glomerular basement membrane is acidic. And so we reasoned that, okay, maybe autoantibodies to DNA can also bind to glomerular basement membrane. And that is how the disease is initiated. And so we set about to test that. We had a large panel of monoclonal antibodies, all of which bound to DNA similarly, some of which cross-reacted with glomerular basement membrane, some didn't. So we had the tools to test our hypothesis. So we did test that test by injecting mice with a pair of antibodies, both of which bound DNA equivalently. One cross-reacted with glomerular basement membrane, one didn't. We could distinguish them because they were separate IgG subclasses. And so what we discovered is that the antibodies were distinct in their ability to cross-react with glomerular basement membrane. So one antibody always bound to glomerular basement membrane, whether alone or incubated with the other antibody. And the other antibody could not bind to glomerular basement membrane. When we analyzed the mice into which these antibodies were injected, well, the result was the cross-reacting antibody bound in the kidney. The non-cross-reacting antibody did not. If we ask where does that antibody bind? Well, it binds along the glomerular basement membrane. Everywhere you see here, where there's turquoise, indicates colocalization of glomerular basement membrane with IgG autoantibody to DNA. So these antibodies are binding directly to the basement membrane. They don't need DNA in the kidney in order to bind. Once they're there, they can also initiate complement activation. And so they can initiate inflammation. When we examined a panel of monoclonal antibodies, similarly, we found that the only thing that was in common with those antibodies that can bind glomeruli and initiate glomerulonephritis was their ability to bind with relatively high affinity to glomerular basement membrane. Binding to DNA, binding to chromatin, binding to nucleosomes did not correlate with their ability to initiate glomerulonephritis. That, by the way, is the transgenic antibody. And that is why the mice that we made transgenic did not develop glomerulonephritis. So, what do we conclude from that? Monoclonal anti-DNA antibodies can bind directly to glomerular basement membrane, and we believe that that is the way that glomerulonephritis is initiated. That also explains serologically positive, clinically quiescent. If the anti-DNA antibody that a patient produces initially doesn't cross-react with glomerular basement membrane, that autoantibody may not be able to initiate the process of glomerulonephritis. And so I'll leave you with this model of how we think the process occurs. We think that the initial event is the autoantibody binding directly to glomerular basement membrane, activates complement. That complement activation initiates inflammation and the attraction of neutrophils and monocytes to that site where, with their Fc receptors for IgG, they can bind to that deposited immunoglobulin that activates them. And part of the activation process of neutrophils, at any rate, is something called netosis, the energy-dependent release of chromatin by those activated cells. So now, if that happens, that chromatin now is available for binding to antibodies that are already bound to the glomerulus, to the glomerular basement membrane. And through this process, which is progressive and cumulative, you generate very large lattice immune complexes that the kidneys can no longer remove. Up to this point, the process is reversible. Mark Manick showed that many, many years ago, that the kidneys have an ability to eliminate stuff that is bound to the glomerular basement membrane to a point. Once the lattice becomes too large, it can't be removed. And that immune complex lattice is what initiates the process of inflammation and kidney destruction. So, here are the people that did the work I just described. Meera Krishnan and Sumyao Wang did the passive immunization studies in mice. Meera is now an award-winning advanced biology high school teacher in Memphis. Sumyao is the vice president of a small private hospital in Beijing. Dharmesh did the experiments on the immunization model for mice. Dharmesh is now a group leader at Bristol Myers. And Meredith did the work on the transgenic mouse, and Meredith is now a senior scientist at Eli Lilly. These are other individuals that have contributed through the years, both as members of my lab and as individuals, colleagues at UT, and this guy is no longer at UT, but we still see each other quite a bit. And then other individuals who have contributed materials and insight to the process, and the NIH for graciously supplying us with money to support the research. So thank you for your attention.
Certainly time for some questions. Can I take the privilege of asking the first one?
I think
so. What are the implications for treatment strategies then for lupus, given what you've described?
Well, I think that the treatment strategies now are by and large ablation. And so you just go in with a big-ass hammer and you kill all the cells, the immune cells. And that hasn't worked particularly well. B-cell ablation therapy just has not been effective. And so I think that the only therapy that's going to be effective here, as we discussed last night, is that we have to reset the immune system. So we have to devise a way to go in and change what is a process of activation to a process that inhibits activation of the specific immune response. Not everything. Don't ablate the entire immune system, but rather find a way to reset the immune response that's autoimmune. And there are a number of ways that can be done now to do that. And that is if we can find a way to engage regulatory T cells to inhibit this response, I think that would work. And I will take the liberty of mentioning something that this is just a thought, an idea, but this is a place for ideas, right? So one of the therapies that is used in lupus now is something called IVIG. And it's just infusing patients with gamma globulin from normal individuals. And what we find is that that reduces autoimmunity and inflammation. Why is not quite understood. It certainly involves Fc receptors and likely involves the ability of Fc receptors engaged to downregulate both B cells and T cells. So what if we take a different approach to that and say take an autoantibody that reacts very well with DNA but can't cause disease, and we infuse that? Would that reset the response so that you change the activation of immune cells that are making autoantibodies that cause disease so that now they don't? Don't know whether that would work or not. We want to try it in mice and see if that's possible. But it's resetting the immune system is going to have to be what solves the problem.
Thank you. I think we had a question at the back. Thank you.
Hi, my name is Greg Lagana. I'm a retired internist, and so lupus was kind of like one of the banes of my existence during my years of active practice. Is it possible now, since I've been out of the fray for a while, to assay for the cross-reacting anti-DNA antibodies, the ones that go after the basement membrane as well as DNA? Can you detect those?
The answer is yes, but it isn't being done.
Okay.
Larry has a question. Larry, you're always allowed. Yeah, Mike.
Given the model that you showed at I've asked you this 1,000 times, and you always tell me it won't work, but I'm going to do it again. Given your model, why couldn't you take the Genentech drug pulmonase, which is DNase, and just, instead of treating the lung for cystic fibrosis, use it systemically to break up those complexes that hurt things at the kidney?
I don't know the answer to that because I don't know that it's ever, I don't know that it's ever been tried. I mean, it's a rational approach. DNase 1 has been used. And what one can show that in mice, you can sometimes have an effect, sometimes not. It hasn't been done in people, as far as I know. And so I guess that the issue is going to be one of internal competition. Who gets there first? And so if the enzyme gets there first, then I think the chances are good. But if something else gets there first, like an antibody, we have natural antibodies that bind to DNA, IgM antibodies, we all have them right now in your circulation that can bind to DNA. And so if those antibodies get to the DNA first, then that may prohibit that method from working. But it seems like it should be worth a try. God knows we need something in lupus, right?
Okay, we've got time for a couple more. One, the guy next to the lady with the turquoise shirt. Very bright, I can see you both.
Hi, my name is Antonio. So my brother actually had lupus, and this is something I've been wondering. When you get lupus, a lot of the times, it goes to sleep once it finished attacking an organ, in this case, a kidney. So most of the times, once you get a transplant, it goes to sleep. In my brother's scenario, it actually had a little bit difference. At one point, the kidneys died. At this point, before this, he had 30% functioning. When the kidneys died, we were expecting that we would have to go through dialysis and everything, and hoping that the lupus would go down. But it actually changed. It, contracting a little bit of what you just mentioned, the kidneys had filtered out, lupus stopped attacking, and the kidneys gained functionality to 60% of it. Then afterwards, it actually went back again, and this was probably, I don't know, maybe a year. So it reacted again. So is there any way, or has there been any tested to fool the lupus that the kidneys is dead for it to stop attacking it?
I mean, therein is the problem. I mean, the approaches that we have are not selective. And therein is the problem. That the antibodies, so the best drugs right now are drugs that affect B cells, B cell development, and their survival. What those drugs don't do a particularly good job of doing is affecting the cells that are already making antibodies that are in bone marrow, right? And so you can eliminate the B cells, but retain cells that are already making autoantibody in the bone marrow. One of the old ways of treating lupus was plasmapheresis. Is you just dump out everything out of the blood except blood cells. And so essentially you reset the circulation of the individual so that there are no longer any IgG antibodies to DNA present. And so then they were fine, those individuals were fine for a while, but eventually the antibodies are produced anew and you get another round of inflammation in the kidneys. There was actually a drug that was created by a company that no longer exists, so it tells you something about the success, that the idea was that this complex of DNA could be infused into an individual, and that would inhibit B cells. Actually, what it turned out that those molecules did was they just absorbed autoantibody to DNA. And so with each round of infusion of the drug, individual got better because the antibodies went away. They were absorbed. But they always came back. And so the drug failed because the premise didn't work. And so I think from what I'm hearing from you, that this is the same thing that may be happening, is that the potential to produce the antibodies has not been changed. You change whether the antibodies are there or not, but not that potential. And as long as the potential to produce the antibodies are there, then you're going to renew the presence of those antibodies and disease pathogenesis. But, okay, I have to say I'm not a physician, so that may be bullshit. I don't know.
Okay, on that note, we'll draw I always like that word. So we'll draw the session to an end. Please thank both Tony and Lucy for their excellent presentations. And we're going to be back here at 5 to 11, please. You have 25 minutes to